We're going to now hear from Jenna Sullivan-Stack, who studies the impact of climate change and disease on sea stars and other organisms that live in coastal tide pools. Her research also helps connect science with policymakers that live in, connect science with policymakers that are making decisions on ocean protection, excuse me. If you want to work with sea stars, sea urchins, sea anemones, or other cool coastal critters, be sure to see how Jenna got to where she is today. So Jenna's gone, she shared her screen, she's got her video on, so go ahead, Jenna, and take it from here.
Hi, Erin, that was an awesome presentation, super interesting. I hope you all on the line enjoyed it as much as I did. I'm Jenna Sullivan-Stack, like Lindsay said. Thank you so much for having me. It's really exciting that you guys are all online. I know it's a tough time to be learning, but it's really nice to be connecting with all of you and hear your questions later. As Lindsay said, I'm a postdoc at Oregon State, and I can talk a little bit more about what that actually is later. But I can give you just a brief background on where I'm coming from. So I actually grew up in Corvallis. Sounds like some of you guys might be here also. And my first word was backpack, which I wouldn't have thought might be relevant to my career path, but maybe it is. I spent a lot of time outside and really, a lot of time in nature, which is one of the benefits of living here in Corvallis. There's lots of really cool things to do around. Got out to the coast some, but just as a interested kid, thinking tide pools were cool, nothing particularly official about it. I also didn't exactly know what I wanted to do with my life until a little bit later as well. But I ended up going to New Hampshire for college, and I didn't study marine. So right now I'm a marine ecologist. I didn't actually work with ocean science or anything like that in college. I actually did some forest ecology, and I got to do a really cool research project that was about wood wasps, which is this kind of scary looking guy on the top right. This is Syrex noctilio, which Erin talked about invasive species a little at the end. This is an invasive wood wasp in New York State, and it drills holes in trees and it deposits its eggs along with the fungus in the hole, and the fungus eats the inside of the tree that allows the little larvae to better digest the wood, and sometimes that can kill the tree. So my project there was trying to figure out if this invasive species is likely to be a big problem in New York or not. So that was really cool to get to be doing science that seemed like it might have an impact and be able to, help better the world. But I wasn't really involved in the ocean too much beyond loving it. Right after college, I worked as a high school teacher in the Marshall Islands, which is in the South Pacific. So I'm not sure if you can see my cursor or not, but here we are in the very top right up in Oregon. Very bottom left is Australia. Marshall Islands are pretty much right in the middle of the Pacific Ocean, basically. It's a coral atoll. So I lived there for a year and I taught high school biology and life science. And a coral atoll, you can see in the middle picture, it's this really skinny strip of coral reef that rises out of the ocean. And people have lived there for a really long time, making their living in this really tough environment, really. So it was a really great experience for me to see how people live in different parts of the world, completely different from Oregon, learn from them. And as you can imagine, the ocean is pretty much everything to them, really important. So I started really thinking about all the impacts that we're having on the ocean and how I could sort of be part of the solution. So next, I moved to Florida. It sounds like most of us are on the West Coast here. And I hadn't been to Florida before I moved there. I hadn't been to the Marshall Islands before I moved there either, which was a little stressful, but turned out great. So I moved to Florida and I worked at a really small nonprofit whose mission is to just bring more people out into the ocean to give them experiences, learning, and understanding how the ocean systems work and how we're impacting them. And the goal is that people, once they've had these really cool experiences with the ocean, no matter what they do with their lives, they might be better stewards of the planet. So that's what I was doing there. I was teaching little kids up to adults about the Gulf of Mexico ecosystems. But I realized I kind of wanted to, the more I learned about the ocean, the more I realized we have to learn. The more you learn about things, that's kind of the way it goes. The more you realize there still is to learn. So I decided I wanted to go back to school to get a little bit more background and expertise, develop some skills in science so that I could maybe make a bigger impact. So that brought me actually right back to where I started in Corvallis. And I went to graduate school at Oregon State. So I finished my PhD, which is what Erin is about to finish in a couple months. I finished that just recently, a year and a half ago or so. And my PhD research, I got to spend a lot of time on the Oregon coast learning about the community of creatures that lives in the intertidal zone. I can talk about that in a second. And now I am not spending as much time on the coast. I'm doing a little bit more work connecting science with policy, but I can also talk about that. So here is my research, what I'm up to when I'm out at the coast. I wish I could ask, I should have set up a poll, but hopefully some of you guys that are from Oregon have been out into the intertidal. This is the area of coastline that's covered when it's high tide and uncovered when it's low tide. And it's a really stressful place for organisms to make a living. As you can imagine, imagining like your house filling up with water for half the day and then draining for the other half of the day, like figuring out strategies to deal with that. You're getting barraged by waves, then you're getting dried out by the sun. It's a really tough place to make a living, but it's a really cool place for scientists to study because everything is happening in a really small area. At low tide, you can go out there and walk out when it's not underwater. And you can see all of the different community members, all of the different creatures, animals, and algae that all live together and see how they're impacting each other. And if you were to do this, for example, in a forest, you'd be, trying to find all of the mountain lions or bears or whatever, and it would be really difficult and take a lot of time. If you work in the intertidal, you can just walk out there and look down, see what's there. And you can take things away, you can put them back, you can take away the stuff they eat, and you can find out a lot about them. So this system has these orange guys are Pisaster ocraceus, which is the species of sea star or starfish, same thing that I study. Scientists like to say sea star just because they're not fish, but everyone knows what you're talking about if you say starfish. And they like to eat these little sessile invertebrates. So invertebrates that don't move around. There's barnacles and mussels here. Their favorite food is mussels. And just to give you, this is a lot, but you guys can do it, I promise. Just to give you an idea of how much we know about the intertidal zone and how much we're learning all the time. This is meant to depict the interactions that are happening in a community that lives in the intertidal. So look at the one on the left. And all of these dots are individual species. And the ones on top, trophic means feeding. So these are the trophic interactions between species. So the dots on the top are predators. The ones in the middle are their prey. So let's say like an herbivore. And then the ones on the bottom might be like algae. So each of these individual lines is an interaction that some scientists have gone out and said, this species eats this species. That is a lot of science that's happened to figure out in this particular intertidal zone who eats who. And then we have different types of interactions in these other areas, like positive non-trophic, like a mutualism. For example, sea anemones and clownfish, that's a mutualism you'll remember from Finding Nemo. Or negative non-trophic, that might be competition for space. So it's not anybody's eating anyone else. It's they're in competition or something like that, that they might be at odds with each other. So the intertidal is a really great place to work. We've made a lot of really cool insights about science studying there. But we don't always have to study all of these interactions. Sometimes we can zero in on the most important ones to find out a lot about the system. And here in Oregon, the really important interaction that's really sets the bar for what happens with the rest of the community is the sea star eating this mussel. So Pisaster eating Mytilus californianus, which are these mussels. And when you have a lot of sea stars and they're eating mussels, they're eating away, there's lots of other diverse creatures that get to hang out in the low zone of the intertidal. The mussels can't move down because the sea stars eat them. The sea stars can't move up because they get too dried out. If you take the sea stars away, what you might expect to see is that the mussels can move down and crowd out all of the other invertebrates. So this is sort of what it looks like in practice. Here's the mussel zone. They can't go up too high because they get dried out. They can't go down too low because sea stars are eating them, and that's where they can be. You might recognize this type of zonation, like when you go to a mountain, you can see there's bands where one type of animal or plant might live, and then there's another band that looks different. So what I worked on is this marine disease that's struck the West Coast all the way up and down the West Coast in 2013 and 2014, where these really important predator sea stars, Pisaster, started getting a disease. It was very mysterious. They got these lesions. They lost their grip on rocks. Sometimes their arms would crawl away from their body, which was really creepy looking. They did these weird twisty things. So sea star wasting disease hit, and it was fast. Like you might see something that resembled a sea star when you first got to a field site or an area of the coast where you're working. And then just a few hours later, it was completely disintegrated. And this is just a map of when it hit in 2013. So sea star wasting has been observed in these red dots, and it's spreading up and down the coast. We're here in Oregon in the middle. So it started in 2014 in Oregon. And a lot of our sea star population, lots of different species, not just Pisaster, died off. So this picture was, you can see this sunflower star was really thick all over this rock, and then it was gone just a few weeks later. So really big thing happening. And the question that scientists have is, what is that going to mean for the rest of the community? Here we have these lines that are indicating, again, interactions between one species and the other. So these ones are trophic interactions. You guys probably remember that is feeding. So the sea star is eating this snail, or it's eating these barnacles. So if we lose this sea star, what might happen to the rest of the system? Some of my questions, like smaller questions that I had, are there's this other sea star here on the right. It's called Leptosterius. It's called, or the drab six-armed star, and it eats a lot of the same things as the big predator Pisaster. So I was wondering, we lost a lot of Pisaster from the disease. Can this drab six-armed star fill that role and start eating more, basically, and getting bigger, and control the mussel population? And the other thing I was interested in is how are these, this is a baby Pisaster here on the right, this really small little light sea star. I'm hoping you can see my cursor. And I was wondering how they are going to help recover the Pisaster population. And this is all in the context of all those changes that are happening in the ocean, like Erin was talking about, ocean acidification, as we emit CO2 into the atmosphere, and then it gets into the water and changes the chemistry and also temperature. So that disease is ongoing, marine or sea star wasting disease. This is what I look like when I'm studying it. So here I am on the top left. I know I'm almost out of time already, but I've got strapped on my backpack what's called a quadrat, which is just a standardization thing that you call, it's very high tech. It's just PVC pipe in a square with some lines. So you can put it down in a section of intertidal zone and count what's there, and you know exactly what area you're counting in. And then you can put it in that same section of intertidal zone later and see what has changed. So you can see that in action in the very bottom picture. I'm in the same outfit, that's my field gear, and a bunch of people are using quadrats to count sea stars and mussels and things like that. The other thing that sometimes we do is we install things into the intertidal. So the top right picture, there's those little cages, and I put some Pisaster, that top predator sea star, in there. I put some of that drab six-armed sea star in there, and they're basically little competition rings to see how they grow in the presence or absence of a competitor. And the bottom picture is a wonderful volunteer that worked with me. It's really hard to install things into the intertidal, because you can picture, if you've been to the coast and seen the big waves that crash on the Oregon coast, there's a lot of stress that you have to really install them well. And so I lost a couple cages from big rocks. That's my friend Skylar diving into a tide pool to pick a cage up that fell off. So you have to put in extra, more than you think you need. So that's kind of like what I was saying earlier, where it's a really cool system to be able to manipulate things and cage things in and exclude things and include things and figure out how the system is working. And to give you an idea of what it's like to be out there, look at this picture. See if you might start to see maybe something cool in there, some little sea stars. They're everywhere. If we were in person, I would ask you how many you could have seen before, but since we're not, I can't tell. But I imagine you didn't see all of these. These are all the babies of the Pisaster, the main predator sea star. And there was a huge baby boom. We lost a lot of the adults from sea star wasting, but we got a big influx of these babies, which are called recruits. Okay, so what's happening? Did we end up having mussels taking over everywhere in the intertidal after we lost the sea stars? Some places yes, some places no. It's kind of an unfolding drama that it's all happening relatively slowly. So we're still trying to figure out exactly what's happening here, but it seems like some places are getting a lot of mussels, other places are not, and it has to do with lots of different factors. I found in my research that adult mussels or adult sea stars are really important, but if you have too many babies, they get in the way and they prevent the adults from being able to control the mussels. And I also found out that it turns out that little drab six-armed sea star probably isn't actually going to get bigger or eat more and save the day. So that's the Cliff Notes version of what I found in my PhD. And now I'm working on, like I said, more about conservation and trying to tie science to decisions that are being made, and specifically about marine protected areas. So we have these in Oregon, and there's lots around the world. These are areas of the ocean that are closed off to some sort of destructive activity that humans might do, like destructive types of fishing and things like that. And my research is helping connect the dots for people so that you know that not all of these marine protected areas are the same. If you can allow some things in one and other things in another. But I'm just trying to communicate the really simple idea that if you protect an area better and you make sure that that protection is working, then you end up with better conservation outcomes. So you might recover fish populations better, for example. So that's a really brief delve into what I'm working on as a postdoc. So after I graduated with my PhD, I'm working for 2 years as a postdoc. It's sort of the training step before you do something like become a professor at a university or something like that. And I just wanted to end by saying the way that I've sort of been thinking about coming through a career in science is to be continually adding to your toolbox with things that are going to help you succeed at what you want to do. So for me, the tools that are most important for my work are writing, working as a team. Those are the main ones. So I'm working together with people all the time. It's really fun, and it works best when everyone is a team player. They're dependable. They do what they say they're going to do. And writing for both scientists, people who are also really excited about the intertidal zone, but also just non-scientists and people in general that are interested in either decision makers that are making these decisions, they're not scientists, or just the general public that wants to know. Statistics or using data, like analyzing data to figure out what the patterns are. Time management. Turns out math is actually important for ecology, so that's a plug for paying attention in math class. There are ways that it matters, even though it's kind of hard to tell from where you're sitting some of the time, I'm sure. Being curious, lots of different tools to add to your toolbox as you go along in your careers. Okay, and there's my contact info at the bottom. Thank you guys so much for listening. I hope you found it interesting, and I'm excited for your questions.
Fantastic, Jenna. Thank you for just insights on your career path and what it's like to be a rocky intertidal ecologist, which is jargon for someone who studies the interactions of tidal animals and with their environment and with themselves, as in the animals themselves. So fantastic. And I love your last slide of all the different skills. That's critical. So if you didn't catch it, participants, go back to our website and view the recording in a couple of weeks, and you'll be able to take notes on that. So we have some great questions from our participants. So if you haven't asked a question yet, go ahead and type it into the Q&A. We'll try to get to as many questions as possible. And so we've got some questions about the sea star wastings. And so to the best of your knowledge and the science out there, what is causing it? And I'm kind of going to combine some questions here. What's causing it? And is it potentially climate change related? Or in things like, yeah. So I'll let you, to the best of your abilities, answer that question.
Yeah, that's a really good question. And it's pretty much the first question everyone wants to know when they hear about sea star wasting, what is causing it. And it's been quite a while since this started. So we've had a lot of really great scientists working on trying to figure out exactly that. I would say it is still a little bit unclear what's caused it. There's been some weird patterns, like if you noticed in that map in the beginning, it started in California and Washington and skipped Oregon, and then it ended up in Oregon about a half a year later. Sometimes there's sea stars dying off right in droves right next to an area where the sea stars are not. So our thought right now is that it is likely a virus, and we're not really sure. So there's viruses in the ocean all the time. There's millions of viruses in a very small amount of ocean. That's normal. And what's not normal is for viruses to be, or for sea stars to be dying in droves like this. So there is something called a densovirus, and there were some researchers in Cornell University who injected some sea stars, that sunflower star with all the arms, injected some with this densovirus, injected others with a heat-killed version of the densovirus, so it wasn't active. And they compared, how many got sick or didn't get sick, and they had some pretty good evidence that that was what was causing. They got sea star wasting-looking symptoms from the sea stars from the active virus and not the non-active virus. So that was some pretty good evidence that that's what's happening. But it might just be for that species. There might be different viruses affecting different species. And it looks like temperature plays a role, which makes logical sense. Warmer waters it's just more likely to have diseases happening faster. So we don't think that warming temperatures is what actually started or is causing the disease, but we think it's really exacerbating it or making it much worse than it would be otherwise. And also when you're really stressed out by a lot of different things, for me, like when I was taking tests and things, and I would study really hard and take a test and be exhausted, that's when I would get sick. So it can be kind of similar for sea stars when they're stressed out from things like temperature or ocean acidification and expending a lot of energy dealing with that, then they can be more susceptible to getting sick. So that's how climate change is likely related. But there's still a lot to figure out about it.
I figured as much. And so, as I said, to the best of your knowledge and the research that's out there so far, we're all continuing to learn about it. And I love your analogy of when you're stressed and you're down, you're more vulnerable to getting sick, right? So that was a great analogy. And so just to be clear, there's no cure. We have a question about, is there a cure for the sea star sickness?
Yeah, so what actually, I think similar to people too, even if it's a virus at first, there can be secondary infection. So after the initial infection by the virus, then you're weakened and you can be susceptible to other stuff. So secondary bacteria infections can be what cause that, turning it into goo symptom. And so people have injected antibiotics into sea stars, and it looks like it does help with those secondary bacteria infections, and also keeping them in colder water keeps them healthier longer. We found that in sea stars and aquaria and things like that. But there isn't really in the ocean. It's so different from on land. The water just makes things happen differently, and there isn't really a feasible way for us to go around injecting sea stars with antibiotics, and it wouldn't necessarily work anyway. So I think the better solution would be to curb our climate change emissions, and I think marine diseases in general are projected to get worse as our climate changes. So I think the best thing to do is to work on greenhouse gas emissions and things like that, and also try and learn more about marine diseases. So.
Great, thank you, Jenna. And we have a great question from Kaylee, maybe channeling her inner scientist. Were there places along the Oregon coast that lost more sea stars? So was it location dependent, the impact? You can even expand if you want to. Oregon, West Coast, I leave it to you.
There definitely was. So there's about, I think it's about between 20 and 30 different species of sea star along the West Coast anyway. So different species got sick more or less than other species. And then also, so in my experience, there's parts of the Oregon coast that started out with way more sea stars than other parts. And that sort of has to do with the life history. So the life cycle of a sea star. I don't know if you guys know this. Oh, hold on. I hope I have time. I think I can do it really fast. I actually have a cool video that I didn't put in here, but I might be able to show you anyway, about what a sea star looks like when it's a very baby, which is not what you probably would expect.
If you're going to show baby
Sorry,
stars, I
Lindsay.
think we might be able to have them hang out with us for a little bit longer, Jenna.
Yeah. Okay, I'm going to share my screen. Okay. Play from current slide. Okay. This might not be the thing that you think of when you think of baby sea star. Do you see it? Is it working?
Oh yeah. It's up in the
Okay,
corner.
so that is yeah, it's up in the corner. That's a sea star larvae. It's called a Brachiolaria larvae.
Whoa.
So you wouldn't expect that would turn into a sea star. So what sea stars do is they broadcast spawn eggs and sperm, and that a fertilized egg becomes a larvae. It starts out like kind of like a blob, but it develops into what's this called a Brachiolaria larvae. And that floats around in the ocean for a couple months. This is a Pisaster sea star larvae. So that purple sea star, the main predator one. It's out in the ocean. That one just hit did you see it went down really fast? It just hit a layer of fresh water. So that's its behavior when it hits fresh water. So they're out in the ocean for a few months, and then they come back into the intertidal zone, or like the tide pool areas, and settle, and they turn into these little tiny microscopic, like they look like little cotton ball poof things. And then they develop into what you guys saw as really small sea stars. I forget where I was going with that. Oh, so there's different parts of the coast that have different numbers of sea stars, and a lot of it depends on the oceanography, or how the currents and how long the water, like how the larvae are moving around out in the ocean, and how likely they are to hit different parts of the coast based on how the currents work. So places that had more sea stars to begin with tended to lose more sea stars, which makes sense. So there's like Cape Perpetua area has a lot of sea star larvae that come in. It also has a lot of larvae of other things like the sea stars like to eat, like mussels and barnacles. And so there were a ton of sea stars there, and we saw a big decline of sea stars there. Other places there weren't as many, and there was a little bit less of a decline, but it sort of, yeah, it depends a lot on where you are.
Fantastic.
Hopefully that helps you.
Thank you. And then I'm going to squeeze in one more quick question. Thanks for hanging out with us, folks. And so we have a great question from Sydney, who looks like she spent some time, researching on her own sea stars. So good job, Sydney. Looks like she's done some projects over the course of the past 5 years. And so great question about how would you compare the population of sea stars from before sea star wasting disease to now? And so if you want to focus on that part, but then there's another part of her question of how higher up predators have been affecting the sea star populations, but separately from wasting disease.
Great. Yeah, so we just had in our lab, so other people that I work with just published a study about that sunflower star. It's called Pycnopodia, and they said 96% decline relative to before sea star wasting. Sorry, there's some baby noises in the background. Hi, bud. So 96% decline in Pycnopodia. And they actually were just listed by the IUCN, the International Union for the Conservation of Nature, on their red list for threatened and endangered species, because they're really worried about the fact that there really aren't any Pycnopodia to be seen, where they used to be really common if you were diving off of the west coast. So it depends, again, on the species. Pisaster, I'm not actually sure. I haven't been out in the field recently, partly because of the baby and partly because of COVID and problems with getting out to the coast. So I'm not really sure what's happening right now, but there was a big pulse of recruits, and I think in some places they're growing up nicely and populations are recovering, but in other places they aren't. So again, I think it sort of depends on where you are.
But real quick, just to correct me if I'm wrong, to get to the part of her question about like what data sets existed before, like how we knew about the populations of sea stars before. PSCO, which I'm going to let you define that. Jenna works in a lab, or worked in a lab that has been monitoring sea star populations and tide pool critter populations for years, correct?
Yeah, so that's a really important point. Thank you. And I'm sorry, I didn't understand the question right.
No, no
In
worries.
order to know what happens when something big, like some big change happens, you have to know it was there in the first place. And that's why it's so important to do the long-term ecological research. So our lab has, for decades has been going out and counting and figuring out the densities of sea stars. PSCO is the Partnership for Interdisciplinary Studies of Coastal Oceans. If you're interested in it, you can look it up. They've got a website. Researchers from that group, including us, have been going out to the same places along the West Coast and doing the same methods to figure out what populations look like for the last decade. So then we have this really long-term information about what it looked like before the sea star wasting disease, and now we can say how it changed after. Whereas if we hadn't done all that work, we would see sea stars dying, but we wouldn't have a way to tell what the actual impact was.
Great, Jenna. And also, Kate, thank you for putting some links into the chat. So participants, there's some great articles that Kate's linking, and also the PSCO Lab website. And just because this is relevant to our participants, is there a way that they can volunteer? You mentioned a volunteer. So how old do you have to be to volunteer for the PSCO Lab? And what does that look like? And I guess COVID might impact that. But just if our participants are interested in gaining some fieldwork experience in those tide pools, how could they get involved?
Yeah, that's great. We love it when people want to come out and help us out and learn about the intertidal. I don't know how that works with COVID. I think we might have to wait a little while on that. It's hard to get out. I think the safety concerns of being out in the field, like often the low tide is really early in the morning, so we're out at dark. And I think adding the pandemic to it is probably, it makes sense to wait till that's all figured out. But once that's figured out, then it's never too early to get out into the intertidal and do some science. So you can contact me, and I can put you in contact with whoever would be the volunteer coordinator. It might change between now and when we're all able to get together again.
But just for our participants to bookmark, they can bookmark the PSCO website that is in the chat and contact the volunteer coordinator if they're interested in getting some fieldwork experience. Is that fair?
Definitely.
All right, that's in the chat, friends. Great. Awesome. Okay, well, thank you so much, participants, for all your great questions. Thanks for sticking it out with us. And thank you, Jenna, for your fantastic presentation and great insights. So thank you for your time, and thank you, participants, for your questions.